find that, and this was even more interesting. Even more interesting from the point, if you withdraw water for 60 days in these plants, the ones which have not received the extra copy of these genes, they were not surviving. Whereas those which had received, they survived 100%. Now if you rewater these plants, they are not able to come up, whereas these plants grow and produce. So we had some indication. This was nice data, nice observations which we got, that this particular pathway, which was discovered to have some relevance to stress tolerance, when manipulated in the crop plant, is possible to develop a new trait within them, the trait of salinity and crop plant. That was right. Of course, these will have to be tested further, but we also looked whether there is any molecular mechanism. You know, I'm basically a fundamental science, basic scientist, but only based on when you find something you want to go into the application. So we're looking into molecular mechanisms, how these are able to talk. How do you get these kinds of root traits which are bigger or smaller into the non-transmitted transgenic? We did some studies on, I'm not going into details, but basically give you an idea that we are moving into those directions. We did a lot of transcriptomics works to find out what are the different sets of genes which are changing. We also did lots of proteomics work to look at the total proteome set which is changing in these plants, and again found some of the genes which are there. But one of the genes or protein which we found was an interesting one. This is a triose phosphate isomerase. So this is the fourth name I am giving you. Methyl glycol, glycolase 1, glycolase 2. The fourth name is triose phosphate isomerase, on which a lot of work has been done in animal systems. So what is this protein? Just give you one slide. Now why I picked up this protein triose phosphate isomerase, because in animal systems it was known, and it was actually associated with some of the neurological diseases, that if there is a mutation in this gene, then there is lot more accumulation of the substrate of methyl glycol, which is dihydroxyacetone phosphate. So dihydroxyacetone phosphate makes methyl glycol, and as I told you, methyl glycol is a very damaging molecule. Oxidative damage, protein modification, DNA modification, and it has to be removed out of the system by this glycolase. So if you have a gene which can regulate even the substrate of this, probably would be very important, because probably in these plants, if the levels of this is more, then the reaction will go in this direction. The substrate would be less and less of the matter. So two ways by which you can regulate the concentration of this particular molecule, by this pathway or by catabolism. That's why in the proteomic studies, when we found this protein, it became very interesting. A lot of more work has been done. One more full PhD thesis has just been submitted on TPI itself and lines, but I'm not going into those details. But when we found that the mechanism by which these plants are tolerating these Mg levels, these stress levels, is because in these transgenic plants you have less of lipid peroxidation. There is a balance of glutathione reduced versus oxidized form. Ascorbate is more. Reactive oxygen species are also slightly increased. Mg level is not increased in the plants which were transgenic. And that probably gives you better survival because antioxidants. Don't worry about it. My major question was that if I'm making more of glycolase, for which Mg is a substrate, the more and more I produce, I should get less and less of this compound. But even many of the transgenic plants which had very high level of glycolase, Mg level was not going down. That means a basal level of Mg somehow is required. That's what I'm saying, even in anything. Some level is very beneficial. You cannot get rid of this compound. So the question was, what is its role, if any? Why this Mg is not there? And then the other question came, does it itself act as a seed? So a lot of work was done, and we found that Mg in fact affects the expression, itself affects the expression of these two enzymes, glycolase 1. This is a substrate-induced expression system, and also many other genes are getting affected. final model which this is where I will end up my work at the moment, is something like this. What we see, this is some new hypothesis on which we are building up. When plants receive stress, there are many signals which are produced in the plants. They have to find they are under stress. So one of the compounds which gives that message to the plants is probably methyl glycol. It's a toxin. So once the level of this methyl glycol increases in the plant system, the plants feel they are under stress. Okay? And they are going to die. So they have to find ways and mechanisms by which you have to protect yourself. So what does this methyl glycol do? It affects this TPI, increases the triphosphate isomerase, and since more of this will decrease the substrate, gives a negative effect, the Mg levels will go down. The second thing this Mg does, it also induces the expression of its own genes, glycosyl and glycosyl. So there is more of these produced which help it in the detox. Two-way strategy. Decrease the substrate and increase the enzyme which will get the glycosyl. And that is probably the initial survival strategy which the plants adopt in order to survive under the stress condition. Now, in order to detoxify, you need glutathione. So what they have also done, this is a beautiful system. This the methyl glycol we found also uses the expression of the enzymes which are involved in glutathione biopsy. So in the process, it increases glutathione. So it collects all the parameters which it needs in order to detoxify the system and survive. However, if the stress persists for a longer period of time, the survival is difficult. And that is why, that's why in the transgenic plant, we try to keep the levels of this very high by different sets of promoters which are constituted. In the normal system, these exist, but the levels are very, very low. So in the transgenic system, what we have basically simply done, allowed the plant to produce more of these proteins so that they can survive. This is a hypothesis which we have did for plant survival. Now, this work which we had been doing was our domain for a very, very long time. Many people did not take it, forget about it, and that we will be safe because many people will not do right. There are only 2 or 3 groups in the world which were working on this psychological practice. It is not even mentioned in biochemistry textbooks. That's a cycle is not too much to talk about. I don't know, many of your biochemistry students may not have been there about it. So we felt safe for some time, but not anymore. Because the groups from America, Japan, and also China, Japan, and US, and one Korean group, they have now started in their own models which they have been working for a long time, how the stress is perceived and regulated. They have started including the stress cycle. And also in the meta tolerance, they now try to put this Mg as one of the numbers. So now we are getting one. The infection is built up. And even I was there, I was talking about Cornelli, which has been working in animal systems. We were called there, and they said the mechanisms by which glycolysis system in aging and stresses work in animal systems, people find that this also works in glycolysis. So more and more people getting involved. And there are a large number of papers. I was telling the area of genomics and proteomics, large number of papers which have come, which now find the relationship of glycolysis bond with one or the other stress tolerance. So the literature is getting flooded with this. And therefore, my suggestion now is that the people will accept to put methyl glycol and glycolysis as stress markers. And these are things that qualify as a stress marker. If we manipulate this, as I was shown, we can develop stress tolerance. And this is a technology which has now been given to a seed company, which have to test under field conditions whether this will work or not, and also do multiple field trials. So that is where we got stuck up in order to we are not getting permissions to do these things. But I hope someday people will realize the value of these times. I'll just take another few minutes.